Research is the cornerstone of innovation in clinical practice, driving improvements in patient outcomes and shaping the future of medicine. Welcome to the Global Rhinology Network Topical Research Symposium. I'm Ashok Rokade from Winchester, UK, and it is my privilege to welcome you as a host. Joining me as co-hosts are Louise Melia from Bath, UK, and Professor Thibault Van Zele from Ghent, Belgium. This symposium showcases fascinating topics across 4 key modules: General Rhinology, COVID session, and Research in Chronic RhinoSinusitis. A special highlight is the PhD session featuring research conducted by enthusiastic young rhinologists who represent the future of our field. This symposium brings together leading global minds in rhinology who will highlight latest developments with a focus on translating research into impactful patient care. The Global Rhinology Network is dedicated to advancing knowledge innovation. Thank you for being part of this exciting journey. Thanks to Medtronic and Karl Storz for their generous support in making this symposium possible. Welcome to GRN Topical Research Symposium.
And we will start with Professor Benjamin Bleier, who is an associate professor at Harvard Medical School and Massachusetts Eye and Ear Institute. He is the director of the translational research. He has published more than 200 peer-reviewed articles and book chapters, and he lectures internationally on endoscopic management of orbit and skull-based tumors, but also on intranasal drug delivery to the brain. And he will talk on the identification and therapeutic targeting of novel epithelial drivers of CRS.
Hi, thanks so much. It's an honor to be here. Let me go ahead and share my screen, make sure we can see everything. All right, can you see my screen there?
Yes,
we can.
well, it's in a slide, not really in slideshow.
How's that?
Yeah, perfect.
Great. All right, well, thanks again. Here are my disclosures, none of which are relevant to the talk. And we're going to be covering some, as noted in the title, some novel upstream potential drivers of inflammation and thinking about how to target them therapeutically. So we know that when we think about the pathophysiology of a variety of types of chronic rhinosinusitis, there is a very complex array and multiple cascades of both type 2 and non-type 2 inflammation. And as we know from the other speakers in this session, the advent of biologics has not only been a clinical benefit to our patients, but also from a scientific perspective, I think it proves that targeting some of these downstream pathways provides significant therapeutic value. But what I find very interesting about these studies, particularly the Dupixent study here, is that when the patient is withdrawn from the medication, the disease returns. And what this tells me as a scientist is that there is something that is still sort of a remnant within the mucosa driving the inflammation that is being masked during treatment, but is still there and present. And this forces me to say, let's look upstream at the epithelium itself to see, are there more upstream drivers of inflammation that we could target that could short circuit the inflammation and prevent this recurrence down the road. Now, there are multiple epithelial derived pathways that have been looked at, and I'm going to focus on 2 for the purposes of today's talk. The first is the role of P-glycoprotein, which is a membrane-based efflux pump, which has been shown to orchestrate and drive cytokine release from epithelial cells. And the second is cystatin, and in particularly cystatin-1, which is a cysteine protease inhibitor, which has also been shown reciprocally to drive epithelial type 2 inflammation. And as you can see from this illustration, that both of these mediators sort of converge on these epithelial type 2 inflammatory cytokines and TSLP in particular, and therefore have some relationship between the two of them in terms of driving upstream inflammation. We'll explore that a little bit. So first, let's focus on P-glycoprotein. Now, this is as I mentioned, a membrane-bound ATP-dependent pump, which is part of the MDR1 gene family. And what this essentially does is when it binds ATP, it takes substrates from the interior of the cell and pumps them out of the cell. So in doing so, it can be considered a pump that is involved in xenobiotic efflux and was actually first discovered in its role in effluxing chemotherapies from the cell. But also it has been shown to be an immunomodulator. It's present on a variety of cells, not just epithelial cells, but also dendritic cells and T cells. And indeed, its expression has been shown to be responsive to the presence of inflammation. And so in some ways, we can think of this as a driver of innate chemoimmunity. Now, we've looked at this protein through over multiple studies in the past decade, and we have shown several interesting findings. Not only have we shown that it's present in sinonasal epithelium, but in fact, that it's overexpressed in the setting of inflammation. And in particular, as you go up in this sort of type 2 inflammatory pathway, we get a higher degree of overexpression. We've also shown that this pump is directly responsible for regulating the release of a variety of cytokines into the surrounding milieu, thereby contributing to sinonasal inflammation. And the concept then is that by inhibiting this pump, the thought would be that potentially we could abrogate some of that inflammation, both epithelial and downstream. And the molecule that we've looked at in the past to affect this is verapamil. Now, verapamil, you may know as a calcium channel blocker, but it was also one of the first, in fact, the first inhibitor of P-glycoprotein discovered in the 1980s. It's considered a first-generation inhibitor because it is relatively dirty, because obviously it has multiple off-target effects. But verapamil is also used in patients, non-cardiac patients who have cluster headache, at a starting dose of 80 milligrams PO 3 times a day. And so from that perspective, we felt that verapamil had a very nice safety profile that could be used as an exploratory therapeutic for the treatment of type 2 sinusitis, and specifically nasal polyps. So in fact, in 2017, we undertook a double-blind placebo-controlled trial looking specifically at oral verapamil for the treatment of nasal polyps. And in this study, we gave 10 patients oral verapamil after a washout period, and then another 10 patients a placebo. And none of these patients were allowed to be on any topical or oral steroids during the course of the trial. The endpoints of this trial, the primary endpoint was SNOT-22. And if you look over the 8-week period, there was no change in the SNOT-22 score over that time in the placebo group, whereas we saw about a 28-point decline in SNOT score over 8 weeks in the verapamil-only group. Now, this correlated nicely with their visual analog scores. And we also looked at some objective measures, including CT scores. So all patients had a CT scan at the end of the study, and the verapamil group had a statistically significant lower Lund-Mackay score. And this is just an example of a patient who happened to have a CT scan prior to enrollment, where you can see pansinusitis, complete opacification. And after 8 weeks of nothing but verapamil, you can see that there was a significant reduction in polyp burden. So again, a nice proof of concept that verapamil acting on PGP was able to abrogate some of the disease state. And over time, PGP has become recognized in the literature as one of the drivers of epithelial inflammation, and that will become important in some of the studies that I'm going to quote later. Now, interestingly, in addition to the fact that PGP seems to have an intrinsic role in orchestrating inflammation, because as an ATP-dependent pump, it's relatively promiscuous, it's also known that PGP regulates the efflux of corticosteroids. And in fact, it had been known that for things like dexamethasone and prednisone were substrates for PGP. But last year, we actually looked specifically at mometasone and found that mometasone also is a substrate for PGP. And what I mean by that is we took nasal polyp explants and we exposed them to mometasone, and we looked over time at how much mometasone was retained within the explants. And we also correlated that with the amount of intrinsic PGP expression within those explants. And what we can see is that as PGP expression goes up, there's a significant and inverse relationship between the amount of mometasone retained within those explants. So in effect, explants that have high PGP levels pump out mometasone back into the environment after exposure. Now, when we inhibit PGP with verapamil, we see no relationship between mometasone retention and PGP, again, suggesting that this effect is mediated through the P-glycoprotein efflux action. When we look at the change in PGP expression after exposure to mometasone or mometasone with verapamil, we see no difference. So again, the effect here is on pump activity, not pump expression. When we look over time at the role of PGP inhibition at retaining mometasone, we can see that after an hour, when we expose explants to mometasone alone or mometasone with verapamil, we see a statistically significant increase in intracellular mometasone retention. So essentially, the introduction of PGP inhibition allows more mometasone retained within that cell. And when we look in these polycellular explants and we look at IL-5 levels, we can actually see that the combination of verapamil and mometasone reduces IL-5 within the explant itself, more so than verapamil or mometasone together as monotherapies, and certainly more statistically significantly more than vehicle alone. So this sort of gives us a sense that P-glycoprotein, again, is acting as a epithelial orchestrator of inflammation, and that the inhibition of P-glycoprotein may be therapeutic, at least in patients with nasal polyps. Now, moving on to cystatin, a lot has been done with this in a variety of labs, including our own. And cystatin is a cysteine protease inhibitor, which is a normal, healthy part of our protective system against environmental proteases. So if we take our own healthy nose and expose it to environmental proteases, which are present in a variety of environmental insults like fungus, allergens, and bacteria, we know that cystatin goes up, and in conjunction with that, type 2 inflammation goes up. When you resolve proteases, what's supposed to happen is that these expression levels go down and you return to a state of health. And this is essentially what we see in seasonal allergic rhinitis. However, what we've found is that in type 2 inflammation, specifically polyps, there is some loss of this feedback inhibition where we get this continuous upregulation of cystatin with a subsequent upregulation of Th2 and type 2 inflammation, resulting in the end state of chronic sinusitis with nasal polyps and type 2 disease. Now, we looked at this in a little more detail, and this was published this year in JACI, looking at the role of cystatin as an upstream initiator. And again, this is what I'm talking about now, is the role of cystatin actually initiating this entire cascade of type 2 inflammation. So this started off by just observationally looking at the amount of cystatin expression in control versus polyp tissue. So by histology, there was certainly quite a bit of cystatin expressed in the epithelial layer. But when we did a multi-omic and proteomic assay, we actually, over 1,300 proteins looked at, cystatin was consistently among the highest upregulated protein relative to control, not only in tissue, but also in mucus-derived exosomes and mucus itself. And so the fact that cystatin showed up as one of the highest upregulated proteins, regardless of the specific substrate we looked at, was quite interesting to us, because typically when you look at these different substrates, you get a different degree of expression of different underlying biomarkers. When we actually compared cystatin expression to other markers of both inflammation and disease, we found statistically significant and positive correlations between cystatin expression and eosinophils within the tissue, as well as cystatin levels and Lund-Mackay scores. Now here we look both at cystatin 1 and cystatin 2, but for the purposes of the rest of the talk, we'll focus on cystatin 1. One of the questions that comes up is what is responsible for this lack of feedback inhibition? And one of the things that we looked at is the degree of post-translational modification. And in fact, we found that relative to controls, there is a significant amount of post-translational modification, both deamidation, acetylation, and methylation of the cystatins within the disease state. However, the functional consequence of this, I think, remains to be elucidated. So we then went on to say, well, if we know that in the clinical state, cystatin is overexpressed and correlates with inflammation, we now wanted to manipulate an animal model in order to study the effects of cystatin on inflammation. And so we established this mouse model of cystatin exposure, where the nasal cavity was exposed to cystatin over time for 5, 10, or 18 days. And then the degree of both cytokine expression as well as Th2 cell infiltration was studied by ELISA and by flow. Now, what we found as far as the cytokine array was that multiple cytokines and as well as eotaxin was upregulated over the 18-day period. But interestingly, so was P-glycoprotein. So now bringing you back to the beginning of the talk, as I mentioned, P-glycoprotein the expression is sensitive to surrounding inflammation. And so here we can see that in a healthy mouse, the mere exposure of the mucosa to cystatin actually induces an upregulation of P-glycoprotein within their mucosa. And then here on flow, we're looking at the amount of Th2 cells infiltrating the mucosa. And we can see that in the cystatin group, there's this actually did not reach statistical significance, but there was a trend towards an increase in infiltration relative to control. Taking the fact that we know that P-glycoprotein regulates cellular release of cytokines, the next step is we took that model and we introduced a silencing RNA to block P-glycoprotein expression. And so this is just our validation study showing that we are able to successfully abrogate P-glycoprotein expression using the siRNA. What we then did was we introduced the cystatin with the siRNA. And what we found is that in a dose response of 0.1 mg per kg up to 1 mg per kg of the silencing of P-glycoprotein, we were able to abrogate the expression of these type 2 cytokines, where red is upregulation and blue is downregulation. So again, taking this model and exposing them to cystatin and then specifically blocking the expression of P-glycoprotein, we were able to essentially block the ability of those cells to induce the downstream type 2 response. So when we go back to our illustration and look at sort of this nexus of P-glycoprotein and cystatin, what we now see is that cystatin itself actually has a direct influence on P-glycoprotein expression, which then in turn influences cytokine expression. So we sort of see this triangle of effect here, where both of these epithelial mediators are conspiring together to drive this type 2 response. So in effect, what we're saying here is that most studies that look at type 2 inflammation look at the patient in the disease state. It looks at the patient's histology, barrier dysfunction, inflammatory profiles, gene expression, and so forth. But when the patient is already activated in that downstream type 2 inflammation, you've lost that initiating event and the ability to resolve that. What we'd like to do is take the healthy mucosa and see what factors drive from health to disease in this chronic state. And our study in the animal model suggests that this sort of collaboration between P-glycoprotein and cystatin may be one of those driving roles. So where are we going in the future? Well, we've started to develop a mouse knock-in model of human cystatin because mice don't intrinsically express cystatin 1. And we have this model now up and running with pups. And so we are actually able to test those. And we're actually able to show that we can get cystatin to intrinsically be expressed in both nasal mucosa and the lower airway over an 8-week period. And we're then able to take that animal model and perform high throughput drug screens, both in the animal and in vitro, where we can take a variety of different potential therapeutic agents against cystatin and measure them for both their ability to block cystatin expression, their intrinsic cytotoxicity, and their potency, so that we can look in these various axes to find the optimal therapeutics that we may want to put back into the model to test as novel agents for the treatment of the disease. So thank you very much.
Thank you, Benjamin, for this very nice presentation. Do you hear me? Yeah.
Yep.
Okay. I have a question for you, Benjamin. Do you think this mechanism also holds true for, let's say, type 2 disease without polyps?
Yeah, that's a good question. So at least from the P-glycoprotein side, we have done studies, histologic studies, where we've looked at controls, patients with eosinophilic non-polyp CRS and polyp CRS. And in fact, we still see a relative overexpression of PGP in the eosinophilic CRS without polyps. So we do see this sort of spectrum of expression from, sort of healthy to polyp. And so I do feel that that there is a role for this in eosinophilic CRS without polyps.
Okay, thank you. Other questions from the panel?
Yeah.
Right.
How would you interfere with that? Or do you have any models already or any ideas on how you would interfere with this cystatin?
With cystatin. Yeah, so in our drug screens, we have started to identify molecules which can block the expression. You know, the question is whether they interact at the post-transcriptional versus post-translational level, whether they interact at the activity, because one of the questions is, how is cystatin driving this, and is that a function of the protease activity or some other non-competitive binding site that's actually driving inflammation? So I think that remains to be seen, but that is the million-dollar question, obviously, is how we can design therapeutics to target that.